Welcome to the BioCore 381 video on genetic linkage and crossing over. Recall in the video on meiosis that we talked about how the independent assortment of homologous chromosomes during meiosis 1 generates variation within gametes. This is because the orientation of the sister chromatids is random, and thus there are many possible arrangements, such as the three shown here. Let's imagine a cell that is heterozygous for three genes, A, B, and C. Each gene has two alleles, big A little a, big B little b, and big C little c. If all three genes are located on different chromosomes, then they will sort independently, yielding 8 different combinations of alleles. But what if all three genes are located on the same chromosome? Does the law of independent assortment still apply? Genes located on the same chromosome are known as syntenic. The law of independent assortment is still valid for some syntenic genes because of a phenomenon called crossing over. Crossing over is the physical exchange of large chunks of DNA between homologous chromosomes during meiosis. It is often depicted as an X between a pair of homologous chromosomes. Note that here I've drawn a simplified version of crossing over. In reality, it occurs between 4 chromatids, which you will see later in the video. But for now, the result is that the maternal and paternal alleles of gene C are switched. Thus, there is enhanced variation within the gametes. In addition, the likelihood that crossing over will occur is dependent on the distance between two genes. Thus, genes A and C are more likely to have a crossover event between each other than genes A and B. If genes A and B are located very close together, they tend to be inherited as a unit. Therefore, Mendel's law of independent assortment fails for this combination. This phenomenon is known as genetic linkage. How can we tell if two genes are linked? Multi-factor crosses can be used to detect genetic linkage because the phenotypic ratios of the F2 progeny do not fit the expected Mendelian ratios. For example, in a 2-factor cross of sweet peas, Bateson and Punnett crossed a true-breeding purple-flowered plant that had long pollen with a plant that had red flowers and round pollen. The first generation, F1, revealed that purple flowers and long pollen were the dominant phenotypes. After self-crossing the F1 plants, they expected to obtain the 4 phenotypic combinations in the Mendelian ratio of 9:3 to 3:1. However, they observed a ratio of 15:1 to 1:4, many more of the original parental phenotypes than they expected, indicating that the two genes responsible for these traits were inherited as a unit. Therefore, gene P for purple flowers and L for long pollen are most likely on the same chromosome. A chi-squared statistical analysis can be used to determine if the observed outcomes are significantly different from the expected Mendelian ones. But how can we tell what the rate of recombination is between these two genes? To do this, we need a cross where there is only a single dominant allele for each gene so that we can track the dominant phenotype easily. To do this, 2 true brooding strains are crossed with each other, then the heterozygous F1 generation is back-crossed with the recessive parent. First, let's consider a situation in which 2 genes are completely linked. In this case, the Punnett square would look something like this, where there are only 2 possible allele combinations. Thus, there are only 2 possible phenotypes. Now let's contrast this with the genes sorting independently. There are 4 possible phenotypes because the dominant alleles, coding for purple flowers and long pollen, can sort independently. But if genes P and L are on the same chromosome, then the phenotypes represented by the middle boxes would be the result of a recombination event. Because we can track the dominant alleles with this type of cross, the recombination frequency can be determined by the phenotypes of the progeny. This is equal to the number of recombinant offspring divided by the total number of offspring multiplied by 100, and is given in map units, mu, or centimorgans, cm. Because recombination frequency is dependent on the distance between 2 genes, it can be used to approximate this distance. But why are map units just an approximation of distance? Let's look at the lower limits of map distance. Imagine a cross with 1,000 offspring. If 2 genes are completely linked, meaning that there are 0 recombinant offspring, the map distance will also be 0. Alternatively, if genes are on different chromosomes, then we would expect half, or 500, of the offspring to have the quote-unquote recombinant phenotype if we obtain the perfect Mendelian ratio. Another way to think about this is that if two genes are on different chromosomes, each allele has a 50-50 chance of being passed on. In fact, if you look at thousands of pairs of genes, you can graph map units relative to recombination frequency. As we showed previously, this upper limit is 50%. If the relationship between recombination frequency and mapped units was linear, then we would expect something like this dotted line. But that's not what happens. To illustrate, I'm going to exchange map units for a physical distance where one map unit equals approximately 1 megabase. The resulting line looks more like this. Since recombination is partially dependent on distance, the left-hand side of the curve represents genes that are located near each other, while the right-hand side represents those that are far apart. In fact, recombination is so common during meiosis that if two genes on the same chromosome are far enough apart, then they behave as if they are on different chromosomes. Finally, note that the curve is below the dashed lines. The further apart two genes are also increases the likelihood that multiple crossovers will occur. A double recombination is not observable through the phenotype, seemingly reducing the recombination frequency. Other factors, such as the presence of recombination hotspots, can also influence recombination frequencies. Let's go over some of the details of crossing over. We'll return to our cell that is heterozygous for 3 syntenic genes after it has finished G1, S, and G2. Crossover occurs during prophase 1 of meiosis. First, the copied chromosomes condense and the homologous chromosomes begin to align through a mechanism that is not completely understood, but that is dependent on sequence homology. The homologous chromosomes begin synapsis, the process that forms the synaptoneal complex, a protein scaffold that organizes the DNA and links sister chromatid pairs along their entire lengths. Crossing over occurs within the synaptonemal complex. There is at least one crossover per tetrad, and likely more. Crossing over is tightly regulated and directed by a host of proteins. Errors such as a crossover between a non-homologous section can lead to potentially deleterious chromosome abnormalities. As prophase progresses, the synaptonemal complex dissociates, leaving behind a chiasma, or a location where crossing over has occurred, named for the Greek letter chi. For simplicity, I'll show a single crossover, though there would likely be more between the homologs. In prometaphase and metaphase, the chiasmata, plural of chiasma, are very important as they keep the homologous pairs of sister chromatids together and help establish the correct connections with the kinetochore microtubules. Finally, when the chiasma resolves, the two recombined dyads can be pulled apart in anaphase 1. In fact, a lack of crossovers can lead to the missegregation of homologous chromosomes at this stage. Let's jump ahead to metaphase 2, which is akin to metaphase during mitosis. Each kinetochore microtubule is now connected to one of the chromatids, allowing them to be pulled apart in anaphase 2. We started this process with a pair of homologous chromosomes with 3 syntenic genes, each with 2 different alleles. After meiosis and crossing over, there are now 4 unique gametes and 2 new combinations of alleles. Thus, the outcome of recombination is increased variation both within the gametes and the resulting offspring.